Dear coordinators, sponsors, fellow participants, and alumni, we are the Kalchenko Research Group, and our project was optimization of medical image perception in multiple modalities using color mapping and AI. Current estimates predict 1 in 10 of all patient deaths in an outpatient setting are the result of diagnostic error, meaning when a radiologist misses a few pixels on a screen, possibly late at night after a long day of viewing dozens upon dozens of unremarkable scans, a patient's life may be put at risk. Now, a hemorrhage, an early sign of a stroke, or a cancerous tumor has gone undiagnosed. Furthermore, diagnostic errors in the field of radiology are as high as 40 million per year globally, 3 to 5% of all the cases they see. This proportion will only continue to increase as imaging technologies become increasingly advanced, requiring non-specialized physicians to rely more and more on radiologists to make their diagnosis.
I want you to compare the left and right sides of the center bar in figure 1. Now look at figure 2. What's the difference between squares A and B? Unless you've seen these optical illusions before, you probably said one side or square was darker, while the other was lighter. You can probably guess what we're about to show you next. Our perception of shadows masks the fact that the center bar is actually one color and that A and B are the same. Our brains can be easily tricked by shadows and surrounding stimuli, especially when the scenes we're assessing are in black and white. Some research groups have attempted to rectify diagnostic error by focusing on education, teaching physicians about their interpretive biases. Others are attempting to develop machine learning and AI software to try and aid radiologists in the perception of lesions. So when we apply color scheme to our optical illusions, it corrects our skewed perception of them. By modifying the MRI or CT images after they've been scanned, different color schemes can help to detect previously unknown or unseen problems in the original scan.
LUTs or lookup tables are a way to color code medical images as shown on the left. The LUTs are generated by manipulating the hue, saturation, and brightness of a color gradient to match the specific color intensities we want to highlight in the images. These factors are summarized in the HSP model, which is presented on the right. Here is an image of a typical brain scan. Can you find the lesion we have manually simulated in the brain? When a LUT is applied and the scan is colorized, does the lesion become any more visible? This method makes it possible to highlight information which otherwise could be hidden in a grayscale image. Therefore, it is a goal to highlight important information, use datasets to train networks in the future, produce LUTs that would not result in information loss, and to be colorblind friendly.
So how do we make them? We divided the creation of LUTs into 3 methods, based on previous research and our perception. LUTs were created to ensure a good overview without distracting a doctor from potential lesions caused by excessive use of intense colors. Editing the LUTs manually allowed us to control which areas of the scan we might want to show, therefore giving us high precision, and it provided some pleasing results. Unfortunately, this method generates LUTs which are unique to one specific image and may not apply to others. Using existing color schemes saved time. However, they did not give us very useful results, as they would often highlight areas with no interest. Once we have a good palette of colors, we tune the hue, saturation, and brightness so that we can relax background colors, usually the darker, and exaggerate the lighter ones to allow for areas in the scan to stand out. Some of the best color pairs are purple and green, indigo and orange, and black and red.
We also created LUTs based on conic sections. That means depending on what cut you make to a cone and the shape it is, you can produce a LUT with different but useful properties. We found that creating parabolas in this cone would produce a symmetrical effect that we wanted. This symmetry would remove outliers and instead focus on gray matter, which is what we are interested in. Then we decided to colorize the scans and highlight the regions we were most interested in. As shown in the image on screen, certain structures are highlighted by this LUT, helping to maximize the information on the scan. We use these 3 methods to generate all of our LUTs. Depending on the LUTs used, you can run into problems. Information can be lost when the scan becomes colorized. Additionally, the new color schemes can be confusing to look at. LUTs that are too bright can make it hard for a doctor to read the scans and even distract them from the problem at hand. With some people having issues perceiving certain colors, it is necessary to pick specific LUTs that amplify the information but minimize these problems.
We had about 50 different LUTs and narrowed our favorites down to 20, each of which was then applied to MRI scans. We then created a survey in order to test how perception of lesions and abnormalities in the scans differed depending on how the image was altered. The survey was 5 questions long and showed some random grayscale images and 3 random images colored by LUTs. Here are illustrated the responses of everyone, on the left in a normal grayscale image, and on the right images colorized by our LUTs. Each red dot corresponds to a click on the image made by a survey taker where they thought that an abnormality may lie. Looking at the colored LUTs image, the data points don't appear to be less scattered compared to the grayscale image, suggesting that this method of medical image manipulation is not effective in reducing diagnostic errors. However, the sample size of the data is too small for this to be conclusive. Future directions would include increasing the sample size of the survey and including radiologists in our sample population in order to produce more accurate results.
We also developed a code that searches the picture for artificial lesions. The idea was that the colorized scan with the most lesions found could arguably have the most effective LUT. There are 2 components to the program. The first is an edge detector. This part converts the color picture to a grayscale image and then highlights the edges. After this, it sets an intensity threshold that filters out the noise, primarily the brain folds. This leaves only the bold edges, including the lesion edges, should the LUT make it clear enough. The second part of the code searches for lesions. It cycles through the pixels with an interval of 20, setting each pixel as the origin. From each origin, it searches for an edge within 200 pixels. It does this repeatedly rotating around the origin. If it cannot find an edge, it moves on to another origin. If it finds edges and returns to the original angle, then it boxes the shape and labels it a lesion. This method showed that the blue-green LUT was the most effective, revealing 5 out of 5 lesions, followed by fire, LUT 13, and blue-yellow with 4 out of 5 each, and red-blue with 3 out of 5. The code is not without flaws. While it finds the lesions, it also marks other regions as lesions, especially the eyes and spinal cord, as they have similar shapes.
So overall, we've looked at different methods of creating color schemes, which can then be applied to black and white medical images. Our preliminary trials show that colorizing medical images does enhance perception and helps people spot abnormalities on brain scans. There's quite a long way to go before this technique can be used in the clinic, but research in this field is progressing very fast today. For our project, future research could include using computer programs, machine learning, or even eye tracking experiments to better understand which of our color scheme is optimal for human perception. We could then test the color schemes with radiologists to see how it can be applied in a clinical context. And one day, hopefully, colorizing medical images could become standard practice in hospitals with agreed conventions for each imaging modality. And with that, we'd like to say a huge thank you to our program sponsors and the program organizers, as well as our mentor, Dr. Slava. Thank you.